Okay, this is for Chapter 11, Section 4, the last section for Chapter 11. It's all about mutations, and we're going to start with gene regulation. Now, gene regulation is the ability of an organism to control which genes are transcribed in response to the environment. And we talked about transcription and translation last time. Now, in prokaryotes, these are cells without a nucleus. These are controlled by operons. Operons are a section of DNA that contains the genes for the proteins that are needed in response to the environment. And the parts of an operon are as follows. It contains an operator, and an operator is simply a segment of DNA that acts as an on-off switch for transcription. If it doesn't need the gene, it'll turn it off. If it does need it, it'll turn it on. Pretty impressive. It also has a promoter, and this is where RNA polymerase binds to DNA. And a regulatory gene we described above. Now, genes for coding proteins are exactly what it says. It's genes needed for coding proteins. Now, an example of this is the E. coli operon. When there is no lactose or glucose present in the environment, it shuts off the genes for breaking these things down. Just shuts it off. Doesn't need them. So when it is present, when they find lactose or glucose, it just switches it back on so it can break down those molecules. That's pretty impressive. If you're not impressed by that, well, I don't know what to tell you, but I find it impressive. And then we have Hox genes. Now, in almost all multicellular organisms, you can find what are known as Hox genes. A Hox gene determines your body plan. Hox genes make sure your eyes go in the right place. You know, you have one on the left side of your nose, one on the right side of your nose, below your eyebrows, above your mouth. Make sure your arms are attached to your shoulders, not your feet. Your feet are attached to legs, not your head. Now, mutations can occur, which we're going to cover next coming up. And there has been mutations in Hox genes observed with fruit flies. Their legs have been where their antennas should be. So where their antennas are on their head, they actually have legs coming out of there because there was a mutation in the Hox gene. But almost every single multicellular organism out there has a Hox gene that helps determine the body plan. All right, so mutations. So we all make mistakes sometimes, and even our body makes mistakes. And we call these mistakes mutations. Now, a mutation is a permanent change that occurs in a cell's DNA. This mutation will be replicated in future cells, which is why it's a permanent change. So if there's an error in copying the DNA, and most of the time, or it can happen all the time, it happens randomly, and there's no way to get it to happen the way you want it to. It's random. There are some things that affect it. But when one cell has a mutation, when it copies itself, it's going to copy that mutation as well. Now, mutations happen all the time, but they are considered rare. Now, that seems a little contradictory. If they happen all the time, how can they be rare? So here's why. Most mutations have no effect on an organism because there are more than one amino acid that codes for the same thing, or more than one nucleotide that codes for the same amino acid, that I should say. Now, in the billions of bases replicated, there may be a few errors, and these two reasons are why they're considered rare. So let's go to the amino acid chart and clarify even more reasons why most mutations have no effect. Now, just to clarify, they can affect organisms, but most mutations have no effect. So remember we started with the start and stop codons, or we started with the start codon and we end with the stop codon. So let's say I have the mRNA AUG that gives me methionine, which says, hey, we're going to start coding a protein. And then I'm going to start making arms. I don't know what the ingredients are for arms. So let's say it's UAU, which gives me tyrosine. And then I need a serine. But this time, well, that's not a good one because that codes for everything. Let's say I need leucine. But this time I get UU, and instead of A, I get something different, like a U. Then that codes for a completely different amino acid. And what happens with mutations that you're going to see in the phenotype, what you physically see is, so say someone, they have two arms, but we don't get a stop codon. We don't get UAA or UAG or even UGA. So say we should be getting UGA that says, hey, stop making arms, but instead we get UGC. We get cytosine. Well, it's just going to keep going and going and going until it reaches that stop codon. But if you go over here, and let's go back to serine, you can use any of these, proline if you want, it doesn't matter. let's use proline because we use serine already. But if I have UUA and there's a mutation, it changes to UAU, it doesn't matter because it still codes for the exact same thing. So in this example, going from UUA to UUU or UUG or UUA, whatever it may be, it codes for the same thing. So we're still going to get the same protein when we put all those amino acids together. So there are types of mutations, and the first one we're going to start with is a point mutation. Now, there are a few types. Now, the first set of mutations are called point mutations. These happen at a single base or nucleotide or a single point in the DNA sequence. Now, remember, DNA is read 3 bases at a time from left to right, and we call those 3 bases codons. When one base or nucleotide is changed for a different letter, we call it substitution. When you hear point mutation, you should think of the term substitution. So let's take a look here. Here's my original sequence. I know this is DNA because I see T's and not U's. So I have T. All right. Everything's looking good. I have T. Uh-oh. It's been substituted for a C. So when it made the copy, it actually changed something. So now my codons are going to read TAA CTG instead of the original, or that was the original sequence. Now it's going to be read TAA CCG, and that may code for something completely different. So there are 3 types of substitution or point mutations. There's missense, which changes one or more amino acids in a protein. The change in one letter of the codon changes the amino acid to something different. We have nonsense, which changes the amino acid to a stop codon early. And this stops the protein making process early. That protein's not going to be fully made because it stopped too soon. And we have silent. The change in the basis still codes for the same amino acid. It's a silent mutation because you don't see anything different. It codes for the same thing. So let's look at this change and see what type it is. So we have CTG, and it got changed to CCG. So let's take a look at that and figure out what type of mutation it is. So our original one was CTG. We got to go DNA to mRNA. So CTG changes to GAG. So we have GAG, which is glutamic acid. Then we have to go for the other one. We have to change CCG to GGC. And we see GGC goes to glycine. So we have glutamic acid and we have glycine. They are 2 different amino acids. We're supposed to get glutamic acid, but there was a change, a mutation, and we went from CTG to CCG. And when we transcribe that, we see we're winding up with 2 different amino acids. We were supposed to get glutamic acid, which is GAG, but we wound up getting GGC, which is glycine. So let's look back at our definitions and find out what type of mutation there is. So there's missense, changes one or more amino acids in a protein. The change in one letter of the codon changes the amino acid to something different. Is that what happened here? Yes, it is. We changed one letter. We had CTG. We changed one letter, the T, to a C, and we wound up with a completely different amino acid. We didn't change it to a stop codon early. We didn't get any of those. And the change in the base still codes for the same amino acid. We see that it doesn't. There are 2 different amino acids it codes for, so it's a missense point mutation. There's also frameshift mutations. Now, frameshift mutations are the next type, and you'll see why it's called this in just a moment. And there are 2 types of frameshift mutations. There is insertion, which sounds exactly what it is. A nucleotide or base is inserted into the sequence. So essentially a new letter, A, C, G, or T, is added between 2 bases. And we also have deletion, which sounds exactly what it is. A nucleotide or base is removed from the sequence. Essentially, a letter has gone missing. So remember how we said the DNA sequence is read 3 bases at a time from left to right and are called codons? Well, the body or the cell doesn't know there's been a mistake. They don't know there's been a mutation. They don't know that a letter has been inserted or that a letter has gone missing. It's still going to read it 3 bases at a time. It will shift the entire reading sequence. And that's why it's called a frameshift mutation. So here we have our original sequence, CAC, TGT, TAC, GGT, and we can see the amino acids that it codes for. Now down here we have CAC. Everything's working out okay. But then we have deletion. A letter has gone missing because we should get TGT. Instead, we have TTT, and we see that this codes for 2 different amino acids. But it's not just that one codon, it's going to mess up the next one because my next 3 letters are ACG when they should be TAC. We see this right here codes for something different as well, and on it goes. So that's why it's called Frameshift. It throws a whole 3 codons at a time from left to right. It throws it off. And the same thing with insertion, you just insert a letter, and we'll be doing that in class. Now, mutations are random and can happen at any time. It's important to note that there are things that can affect DNA, such as radiation. Mutations can happen without things affecting DNA too, though. Frameshift mutations more adversely affect an organism than point mutations because it throws the entire reading sequence off. Now, mutations can be associated with diseases or disorder. An example is alkaptonuria. Now, people with this can't digest the amino acids phenylalanine, and this discolors the urine. People with this have a high amount of frameshift mutations and missense mutations as well. Now, remind there about missense. Changes 1 or more amino acids in a protein. The change in 1 letter of the codon changes the amino acid to something different. And when you start changing amino acids, you're going to wind up with a different protein. In addition to all these, you also have chromosomal mutations, which happen on chromosomes. Now, these are pretty easy. These are not very difficult at all. Don't confuse them with point or frameshift mutations. So here we have some information, A, B, C, D, E, F, and here we have A, C, D, E, F. So something has gone missing. We call that deletion. Over here, we have A, B, C, D, E, F. Over here, A, B, B, C, D, E, F. This time it's called duplication. We haven't inserted a new letter. We've simply duplicated the information. So that's why it's A, B, B, C, D, E, F. Then over here, we have A, B, C, D, E, F. And what's happened is it comes out AED, C, B, F. It's coming out in order, and that's called inversion. It's going backwards, AED, C, B, F. So the ED, C, and B have been inverted. And over here, the only type that requires 2 chromosomes, we see A, B, C, D, E, F, G, H, I, J, K, L. Over here, we have A, B, C, J, K, L. They flip-flop places, and that is called translocation. Those are the 4 type of chromosome mutations. So really, the only ones you really need to look out for are inversion and translocation. If you can remember, translocation requires 2. You're off to a good start. Deletion, pretty obvious. 1 gets deleted. Duplication, do not confuse that with insertion. We have duplicated an existing letter already, and then inversion, it gets flip-flopped. That will be on your test for sure. So mutation checks. So point mutations can affect an organism too, and this happens with sickle cell. The codon, GAG, codes for glutamic acid, and it's already been converted to mRNA, is replaced with GTG, which is valine. The one change in this one amino acid changes the shape of the red blood cell. So what type of point mutation is this? It is missense because changing 1 letter has changed the amino acid, which in turn changes the protein. Now, does our body check for mutations or even proofread what's going on? Of course it does. About 1 in 100,000 bases has a mutation. It goes unfixed, though, in less than 1 in 1 billion bases. So it goes unfixed in less than 1 in 1 billion. And when you're getting 999,999 letters right, and you're only screwing up 1, that's some pretty good accuracy, but the accuracy can cause a major change in the genotype and the phenotype. So remember how we said some substances can help cause mutations like radiation? We call these mutagens. All right, mutation or death of a cell. So a somatic cell is a body cell, which is why they do not get passed on to the next generation, because your body cells are 2 end cells or diploid cells, and they're not involved in offspring or sexual reproduction at all. Now, a somatic cell will affect all future daughter cells, but not your daughters or your sons, just the new existing cells in your body from the point of the mutation onward, unless the mutation has affected the cell cycle, causing cell death. If a mutation is passed on to the next generation, then it's called a germline cell, which simply means the gamete cell, the sperm or egg cell, has a mutation. Most do not affect the function of cells in the offspring, but will only affect the phenotypic characteristics. Now, the phenotype is what you can physically see. In other words, a body cell, which we call a somatic cell with a mutation, does not affect an offspring. A germline, which is a gamete cell, sperm or egg, a sex cell with a mutation, does affect the offspring, because your sex cells or your gamete cells are involved in sexual reproduction, while your body cells are involved in skin repair and mitotic cell divisions with mitosis, while your gamete cells are involved with meiosis. So when you hear germline, we're talking about your offspring's going to have it because that mutation is carried inside the gamete cell. If we're talking about a somatic cell, it will not get passed on to your offspring because we're talking about a body cell that is a diploid cell that has a mutation that's not involved in sexual reproduction. That's going to be it for Chapter 11. If you have any questions, bring them to class, and we'll see you guys next time.